about time we could get started. More people will trickle in, as they usually do. It's my pleasure to introduce Annalisa Berta, who's been a colleague of mine for a long time. We knew each other when I was in graduate school.
Yeah, since the 70s, Blair. No, it was 80s.
Yeah, right. So she got her PhD in 1979, and then she did a postdoc in Florida, at the University of Florida, at the Florida State Museum for a couple of years, and then began her professional academic career at San Diego State in 1982, where she has been since then. And early on, Annalisa worked on carnivores, terrestrial carnivores, that is, and we overlapped in research interests, but I guess I scared her away or something. Maybe not.
It's those saber teeth.
Yeah, it was about 1989 she started working on pinnipeds and then cetaceans, and has been a devoted cetacean pinniped paleobiologist and biologist ever since then. She has over 50 publications on the marine mammals. She has a book that she first published in 1999, Marine Mammals, Evolutionary Biology. It's now, you're working on the third edition, right?
Coming out?
Yeah.
You'll see a promo.
Promo to be mentioned. Yeah, and she's worked on systematics of cetaceans and pinnipeds and marine mammals, systematics, functional morphology, a little bit of feeding ecology, all kinds of things. And today, and this I'm looking forward to hearing about, because we're going to hear some of the first morphometric or whatever analyses of baleen. We're going to hear about new insights on the systematics, anatomy, and evolution of baleen whales. Annalisa.
Okay. Thanks, Blair. I've known Blair and Bob for forever.
Yeah.
And I really, it's true. I met them when they were grad students. And I always wanted to say it was the 70s, and as I tell my students, and some of us remember the 70s. Yes. But in your case, I know you're not that old. It probably was the 80s. But anywhere, somewhere in there.
80s.
Oh, there you are, Bob. Okay, 80s. Okay, so research in my lab, as Blair says, involves systematics, and I'm a morphologist. I also do work with molecular systematists, so I cross over to the other side. Lots of students in my lab, and a number of collaborators, John Gatesy at Riverside, Tom Demeray, who was a former PhD student of Blair's a number of years ago, and then we had a postdoc, Eric Eckdale. So one area of my research is systematics, and then another area is anatomy and evolution. In particular, we've looked at cranial anatomy and evolution of feeding in pinnipeds and cetaceans. We've done osteology, dissection, CT work, histology, using comparative phylogenetic methods. And these are just showing you some of the students in the lab, and I know Bob will remember this guy. Will Airy is just finishing master's degree. He was involved in working, I guess, in your lab for a little bit of time. All right, so today what I want to do is start out telling you a little bit about whales, baleen whales in particular, and phylogeny, look at higher level relationships, and then family and species level. Talk about morphology work, and again, that's mostly what I've done, but again, combining with molecular data, and tell you what the future is with our phylogenetic tree. Then I want to move on to the second part of the talk, where I'll talk about feeding anatomy and talk in particular about some research I've been doing recently on this transition from teeth to baleen. Say a little bit about tooth genes, anatomy of baleen, and then blood supply to baleen, and then finally finish up with what I think is a more integrated way of looking at feeding strategies within the context of a phylogenetic framework. Okay, so for those of you that aren't aware of it, whales here are nested very firmly within even-toed ungulates. Their closest relatives is this group here, the Rheoelid artiodactyls. This superorder is now recognized as Seat artiodactyla. So the group that you'll be hearing a lot more about in my talk are the mysticetes, or the baleen whales, and you can see here that they're much less diverse than the second group here, the odontocetes, or the toothed whales. And so I'll be talking about stem taxa and crown groups. So the crown groups include the extant families as well as their close relatives. The stem taxa are all the fossils that are outside of the crown group. So mysticetes themselves are named for these epidermal plates of keratin that hang down from the upper jaw and function in filter feeding. But very importantly, as you'll see, stem mysticetes and fetal mysticetes actually possess teeth. So there's more to that story. Okay, so in terms of the crown mysticetes, there's 4 families recognized, balenids, 4 species. These include the right whales and the bowhead. The most diverse group are the balanopterids. These are the humpbacks, fin and blue whales. And then we have 2 monotypic groups, the pygmy right whale, Capiria, and Eschrictidae, represented here by the gray whale. So this is some work I did in collaboration with Tom Demeray at the Natural History Museum in San Diego. Rocky McGowan was a master's student of mine and a PhD student of John Gates's, and then John's at UC Riverside. So this was done a little more than 5 years ago, and this is our take on mysticete phylogeny. Most of the data, as you can see here, is this very rich molecular framework, and we had 100 morphological characters. You can see a large number of characters from all different parts of the anatomy of the whale. But again, you'll see that there's many more gene character data, and 27,000 characters, of which a very small fraction are actually parsimony informative. This is the results of our combined analysis, and I'm going to show you the parsimony results, but the Bayesian results are pretty much congruent with this. The numbers here reflect bootstrap values. We looked at 10 extant, 11 extant, and 20 extinct taxa. So let me see if I can get the animation here. So these are the toothed whales. Everything else, so in blue is the toothed whales. Everything else on there is baleen whales. You can see we have the fossils as well as the extant groups. So really I want to call your attention to this toothed group of stem mysticetes. So these are the baleen whales. And then in purple there are the edentulous, or the baleen-bearing mysticetes. Many fossils in there as well as the modern members of the group. And you can see by looking at these bootstrap values that, well, parts of the trees is pretty well supported, but parts of it aren't. So this was sort of a state of the picture here in this 2008 paper. This is a group that we've now come to focus on. It's kind of a wastebasket group of extinct group of cetatheres, but you can see because they're very closely related to the modern species up here, that the position of these guys and figuring out the relationships of these is going to be quite critical to the phylogeny of the extant. This is just to show you that we've done some new work. I had a master's student who just is now getting his PhD in Mark Clemens' lab. But anyway, he worked on right whale phylogeny, and just to he did it using morphological characters, so we had a good suite of characters. Really, this was the most comprehensive phylogeny to date. The main thing to take away from this, balenids are monophyletic, and then there's the right whale group is united with a fossil taxon, and the bowhead is united with another fossil. So their relationships are less well resolved in here, but there actually are 2 separate subclades. So that was done fairly recently. But there are really 2 remaining sort of challenges to mysticete phylogeny, and 2 I'm going to go through. And one of them is the conflict in position of 2 of the crown taxa, pygmy right whale here, and gray whales, and then this enigmatic group, the cetatheres, which are stem or perhaps crown mysticetes. So these are where some of the challenges lie. And I'll kind of introduce you to where the issues are. And this is the pygmy right whale, alternate positions based on whether you're looking at morphology or molecular data. So morphology very squarely puts right whales and pygmy right whales together as a monophyletic group. You'll see the molecular work and combined work, where you look at morphology and the molecules together, actually puts the pygmy right whale as sister to all of the other mysticetes. So a real conflict in terms of position. Now to look at this extinct group, the cetatheres, again, there's controversy about where the cetatheres belong. Are they out? There's some hypotheses that put them outside of the crown, and then there's a couple of other hypotheses that place them in different positions within the crown. So this is an unresolved controversy. This group of cetatheres, this is what we had and I'm very sad to say this was the end of my NSF grant. I hope I get another one, but I don't know the way it's going. But we had a very large grant to kind of work on this group. It's, as I said, it's a wastebasket group with lots of genera and species. The only reason they're kind of lumped together is because they lack synapomorphies of the extant mysticetes. They have quite a large range, 32 million years ago, and there's a recent suggestion that maybe cenotheres still survive today, and I'll get to that in a second, but they have quite a bit of morphological diversity within the group. So this is a newer phylogeny done by Ewan Fordyce, a New Zealand paleontologist, and his graduate student Felix Marx. And I'm going to come back to talk about this a little bit later. But notice here that this is where they are suggesting that a cetothere, Herpetocetus, actually is a surviving lineage today because it's grouped very closely with this pygmy right whale. Okay, so again, this is the idea behind this last survivor of an extinct lineage based on their morphologic phylogeny. Now, Eschrictids, alternate phylogenies, the morphological data has Eschrictids as distinct from Balanopterus species. And actually these are, of course, these are monophyletic and they're called Balanopteroidea, but that's another point, doesn't really matter here. The molecular data, though, has a different topology here where they have actually the gray whale as most closely related to a species of Balanopterid, which is rendering this genus as paraphyletic. But the bottom line is lots of molecular data, like I said, but the addition of only 115 morphological characters overturns this hypothesis of the molecular data that's based on over 30,000 molecular characters. So what we came into this, our focus here was on this extinct group of cenotheres, so our study in progress, I'm going to tell you a little more about, includes the largest number of extinct and extant mysticetes. So really we more than doubled our 2008 paper, and we also doubled the morphological characters from that paper. So we did we're just in this is very preliminary data, but we looked, did ordered, unordered, and analysis of characters. And I'm just going to show you one of the results here. This particular results puts, where the red dot is, that's putting the cenotheres in the crown group. And there's just a couple of points to take away from this. Probably the most important is recall that here was the pygmy right whale, okay? And in that previous hypothesis, it was one of, was closely related to this extinct group of cenotheres. So in order to get them close together using our data, you would have to reverse a large number of characters. There's some good support for some of the basal nodes, but there's not such good support for some of these other nodes here. So that's kind of the takeaway there. And you can see here the gray whale is in this position. So I'll come back and say a little bit more about it. Sort of a summary of this is that our conclusion is that cenotheres are paraphyletic. We confirmed that. And we have them inside the crown, so they're inside with the extant mysticetes, but this result is weakly supported. And our preliminary results define a monophyletic cetothere, but there's another clade of them that's outside this group, so of course that's the reason they're paraphyletic. And finally, we had no support for the pygmy right whale as a cetothere. Okay, so just a little bit of a summary and maybe what could we, what should we do next here in terms of resolving the phylogeny? As I said, this phylogenetic framework is still in progress because we're trying to add a number of important fossil specimens. And then also, as I've shown you, there's conflicting positions of some taxa. So we want to, I don't just want to say that we're just going to expand the taxon sampling or we're just going to look at more characters, because everybody always says that. But it's true. We're going to look at a little bit more of that. But we need to do more sophisticated analyses. It's very clear in the runs of the data that we have had so far when we do these combined analyses that some of these fossil taxa could be defined as rogue taxa. They move in different places on the tree. So maybe kind of pulling some of those out and then rerunning the tree. We haven't gotten to that point yet, but I think would be maybe a step toward some resolution, because there's clearly some sort of signal problems here. And I always say this to my classes. I know I'm not, I'm preaching to the converted already here, but I always say, it's not all about genomic data. I know there's lots of it, but there is a lot of valuable information that can come when you include, if you have a fossil record for a group, you need to include the paleontologic data because, of course, it's not just important just for the relationships, but the character mapping, the divergence, diversification data. So obviously, I'm a paleontologist. Fossils make a difference. All right, so let me transition to the next part of the talk, which is now going to use this phylogenetic framework and really look at this, what I think is one of the most incredible, well, I would call it a macroevolutionary transition, and that's the transition from teeth to baleen. So you're looking here at a fossil taxon with well-developed teeth, and then here is the modern mysticete that lacks teeth and has these baleen plates that it uses to filter feed. Okay, so now the interesting story is that actually baleen whales start out with teeth. The teeth are resorbed before birth. So they go through all of the different stages of teeth. They're mineralized. They're not well mineralized, but they are mineralized. So you can see these well-developed tooth buds. So what we did was we looked at some fetal skulls, and we found evidence of the fact, when you look at the skulls, osteological evidence of the fact that they had teeth. In the adults, you would expect the baleen. You don't see any trace of the teeth, but here you've got these alveolar grooves where the teeth would have been, both in the lower jaw here and the upper jaw. So evidence for teeth, open alveolar groove. Now, if we look at fetal skulls, also in the Smithsonian collections, just a little bit older, but you can start to see the alveolar groove closing up here, right? And then something important here is you'll start to see the development of foramina on the palate. And I'm going to come back and have more to say about that. So really the teeth are lost and the baleen develops. They're almost coincident with one another. And one of the interesting aspects of all of this that we really need to do a whole lot more work with is really the development of baleen. And so there would be some really interesting genomic types of studies you could do. Difficult to find the specimens, however, to do that work on. Many of these specimens the fetal material is in collections, and it's preserved in formalin, which I understand not good for trying to recover any kind of transfer RNA or anything like that. Okay, so this is another part of the same 2008 paper. This was work that I was involved with. Mostly it's work done by a molecular systematist, John Gatesy at Riverside. So what he did was to look specifically at tooth genes. And so he actually sequenced 3 different tooth genes. And then you can see here what happens when you have a mutation of these tooth genes. What it causes is a defect in the development of the teeth. So they get, they're kind of fragile and they're not very well mineralized. Okay? So we made the prediction that enamel-specific genes would be present in the baleen whale genome because, of course, their common ancestors, right, had teeth. So that's a hypothesis. But these genes would be non-functional, right? So they'd be pseudogenes. And actually, this is what John's work discovered. The red bars here, really, these are mysticetes in the purple, and then the red bars, what those refer to are the frame shift mutations. So again, disruption in the protein coding region. And again, you can notice that happens only within the mysticete genome. Okay, so moving on, just a couple of years ago, there was a live stranding of a gray whale, and my students went to Moss Landing and picked the specimen up, brought it back to San Diego State. And the specimen itself, we just took the head. They had use for the other part of the body. It went to another collection. And so this was an anatomical anatomy meeting a couple of years ago. We had Joy Reidenberg, Larry Whitmer, and Tom Demeray here, some other people, students. So we did this dissection of this gray whale head, and because these mysticetes are so large, they're difficult to study in the flesh. I mean, if you want to do a dissection of one, so it's opportunistically that you are able to do a dissection. And so we actually got accepted 6 papers that are going to come out here, and probably after Blair's volume here, an anatomical record, I don't know, maybe in the next year. And so I'll talk a little bit about the research. But here we were interested in some of the details of the baleen itself and the feeding anatomy. Okay, so here's this head, and you can see here you're looking at the palate, and you can see the baleen plates here, so you can see they line the lateral margins of the palate. And when you actually, if you peel back, this is what you're looking at, is a little section here. If you peel back the baleen, you can see that it's very richly vascularized, okay? So it's all these dermal papillae here. So there's a very rich blood supply to the baleen that nourishes it as it grows. I had another student that recently did a master's project on morphometrics of baleen, comparative anatomy of baleen. So you can see here, baleen grows throughout life of the individual, and you can see in the neonate particularly, you can see the well-developed laminae. And these laminae or layers come together as like pages of a book, and they form these racks. So what we did was, because many times in a museum, you just see a couple of isolated, pieces. And so we really needed to get an understanding of the entire rack here. And so she was able to find in collections several racks, and then she did, I think, yeah, here we go. She did some morphometrics to try to look at different, adult, yearling, and neonates, and kind of see if there's some sort of patterns with all of these different measurements. So on the X is number of lamina. Lamina make up plates, and then the plates make up a whole rack, and then just different metrics. So the take-home message here is that the longest, widest, and thickest plates are toward the posterior rack. And this is the little bit of studying that's been done in terms of filtration and how it works on these, very little work, but that's the area of greatest filtration. I had another student that also did a master's degree. He's just finishing up now, but he did more of a histology study of the baleen itself. I'm not going to go through the details here, but just these are, again, it's epithelial tissue. You can recognize the different layers of the epithelium. You can see the baleen itself forms these tubules here, and then he has been able to make some comparisons of this gray whale here with some of the very limited data on some of the other taxa. He's done some X-ray work where you can actually see that there's some calcification in certain parts of the baleen, and it seems to, these guys gray whales feed very differently than some of the other baleen whales, so there's still more to that story that hasn't been told. Okay, so now we're going to move back to the fossils and look at when we look at the fossils, what kind of evidence do we have for baleen in them? And when Tom and I were describing this as a stem mysticete, a fossil, you can see well-developed teeth. But if you look really carefully, and I know there's a bunch of other cracks, but the little sulci that are in red there, we hypothesized that, well, maybe those are the lateral foramina and sulci that perhaps were for blood vessels that nourish the baleen. So that was a hypothesis, and here's what you can see where we got the idea is when you look at a minke whale. And actually, if you look at any baleen whale, they have very different patterns of these lateral foramina. And so we hypothesized that these, in fact, were these lateral foramina on the extinct stem mysticete. Perhaps we hypothesized those as osteological correlates for lateral sulci in the extant taxon. So again, the point is they would have nourished, provided a conduit for blood to get to the baleen.
So they have baleen empty.
Yes, that's it. That's it, Blair. You anticipated. So now we have, yes, beautiful. She didn't see the slides beforehand, I can tell you that. Well, this is of course we have no conjecture, totally artist's idea of what the baleen may have looked like. We have no idea. But we wanted to kind of explore this whole idea a little bit more. And so with this recent stranding of the gray whale, we were able to do some CT work. And so we did some latex injections. And so the latex, and it would be barium the isotopes or the barium would show up on the X-rays as these bright spots. So this is going to give us a little bit more information on now the blood supply. And so we were able to, this is just a CT scan, reconstruction here, but we were actually able to demonstrate that, well, it's true that right next to the baleen are these bony canals that kind of lead into the baleen. And then when we had, with our latex, we were able to inject, it worked, first time we ever tried it, actually worked, so that we were able to actually show what the blood supply was to the baleen and superior alveolar arteries. Now it's not quite, I'm kind of cutting to the chase here on this because it could be a very long story and we don't want to be here all day. But there was another hypothesis had suggested that there might be another series of vessels that would be supplying the baleen. So our study of this gray whale through the latex injection was actually able to confirm that in fact it's the superior alveolar arteries that are supplying the baleen, not any of the greater palatine arteries. Although there are certainly foramina in the midline of the palate as well as on the lateral edges where the baleen is. Okay, so then we kind of looked around and we said, well, God, is there anything else we can look at that has something similar to this? And well, baleen, of course, I didn't mention this, but it's a neomorphic structure. There's obviously nothing else that has it. But the platypus has kind of a similar situation where the teeth of a fetus are resorbed before birth, and so they end up with these green keratin pads. Okay, so perhaps this is analogous in some way to the baleen. And then if you look at the fossil record of the platypus, you see that, wow, look, they've got teeth and they've got these keratinous pads, which is kind of our idea of the stem mysticetes, or some of them having both teeth and baleen. So this is a possibility. Of course, a platypus, not at all closely related to cetaceans, but I think, and maybe Blair can help me out later with this, but I think we probably, artiodactyls have a palatal configuration where they have rough rugose areas of the palate, lack of teeth, sometimes in the upper jaw. So there might be something that's and artiodactyls, of course, are cetaceans are embedded within them. So we might have more luck if we actually look there, but they certainly don't have this same sort of setup. Okay, so just a little summary of this. So anatomy and genetic data, evidence of tooth loss, I've showed you, replacement with baleen. These lateral foramina on the palate, osteological correlates of baleen. And then as I've showed, evidence of pseudogenes in the mystacete genome, which gives you or confirms the toothed heritage of the group. And then now we've been able, through dissection and injection work, to confirm the blood supply to baleen and to note that it is the superior alveolar arteries. Okay, so then the last part of the talk here, class. See, I'm not even, and my class has been over for 2 weeks. Yeah. But one other area that Blair is very involved in feeding anatomy, among other things. So we've added a little bit of work on bulk filter feeding. So what is it? Was there a bulk filter feeding niche in the past? And when did it evolve in mysticetes? What was the ancestral feeding strategy? So first, this slide illustrates that indeed there were other occupants of this bulk filter feeding niche in the past. And during the Mesozoic, there were pachycormid fishes. These are ray fin fishes. And there were also, after that time, in the Cenozoic here, there were a number of basking sharks and other types of sharks, cartilaginous fish here, that were in that filter feeding niche. So yes, there was that niche in the past, and there has been a little bit of work in terms of the biomechanics behind the feeding, and there are some similarities among some of the sharks and mysticetes. So now let me just tell you a little bit about the feeding strategies that we see. They're very different. The skim feeders are the right whales. And they basically move through the water very slowly with their mouths open, sort of continuously feeding on the smallest fraction of zooplankton. They mainly feed on copepods. Engulfment feeders are the fin whales and the humpback, and what they do is they lunge feed. So they engulf large, they go and swim very rapidly through the water and take a mouthful of food and then process it. And in order to process and take in a large amount of water, they have these ventral throat grooves here that I'll show you in a second. And then the third main feeding type is just seen in the eschrictids. I've written here benthic suction feeding, but it's really, as you'll see, a lateral suction feeding, so it's a more specialized kind of suction feeding. And they feed, they feed certainly on fish to some degree, but largely they feed on benthic amphipods. So 3 different feeding strategies. Let me see if I can get this. It's always a gamble with this. This is from the Planet Earth BBC. Let me see if I can. It's just going to show you this is the evolution of mysticete bulk feeding. It's been described as the largest, biomechanical event because it involves them taking in enormous amounts of water. I don't know. There we go. So you're just going to see and you see the throat pleats. That's really all you get out of this, is so they take in, as I said, one gulp, an enormous amount of water. And we're learning a lot more about how they feed because they've been very successful in terms of getting digital tags on these animals. And I mean, they just need to get a camera in the mouth. That hasn't happened yet, but I'm sure that's going to be next. Okay, so if I back up a little bit and tell you about now feeding in these stem tooth mysticetes. Okay, so you can see here that some of these mysticetes, when they start out, some of them are small, some of them are large. Actually, there's been some work here at UCLA, and when Graham Slater, was he a postdoc here in the Alfaro lab?
Yes.
Yeah, so they've looked at clade diversification, and they used whales as an example to kind of look at niche partitioning. And indeed, they discovered that early on, some of these mysticetes were varied sizes, small, medium, and large, and fed very differently. These guys, as you can see here, well-developed teeth. So they were probably suction feeders, single prey suction feeders. This is Adiocetus, the one that Tom Demery and I have worked quite a bit on the systematics of the group. Kind of importantly here for you guys is that this is the taxon that we're suggesting, that this is the stem mysticete that perhaps had baleen as well as teeth. And by that token, could be argued to be the earliest bulk filter feeder within the baleen whales. So now this is some later work, actually published not too long ago, a paper that really John Gatesy, the molecular systematist, was the lead on. And what he did here was to optimize some of the feeding characters onto the tree. And I'll show you just that there's lots of different characters that we're looking at. We're just really, I think we're going to be able to partition these feeding strategies out much, much better. We just kind of know about them in a rather gross way. So we can say that, early on there's suction feeding going on, there's single prey, bulk feeding, perhaps that evolved here within some of these tooth mysticetes. And then here's some of the characters. And then within the crown mysticetes, like the humpback and so forth, you see some of the very derived feeding adaptations where they have these throat pouches, very large size, so they can, again, take in a lot of prey. So this is just a beginning step in terms of looking at feeding strategies. This was a master's project done by one of my students a few years ago. Notice this is a dissection she was at, and she very carefully made sure that she wore her SPAM t-shirt. But this is an archive, so this is available on the web. You've probably you're familiar with this website. Lots of really cool photos and videos of animals. I'm going to just get to it. So you can see this is a gray whale, how they actually go down. They laterally suction feed. They go down to the right side. We can tell that the baleen is worn on that side. You can see here, they don't have the throat pleats, the really numerous throat pleats that we see in the Balanopterids, but they've got a few throat pleats that contribute to suction. Okay, this is part of Cassie's Johnson's study was to do some PCAs and look at suction feeding. She looked at it not just in the gray whale, but in other whales, and so she did a PCA. These are just different characters of the skull, and especially of the hyoid apparatus, because of course there's lots of tongue muscles that attach onto the hyoid, and lots very important in suction feeding. So, basically, she was able to partition the first PC, kind of partition things. Here's the mystocetes over here, here's the odontocetes, so it's sort of a phylogenetic partition, as is this one for the most part as well. And then when you look at PC 2 and 3, it gets to be a little messier with, there's lots of overlap between the different odontocete and mystocete suction feeders. The one other thing to point out that it turns out that suction feeding's we know it's more complex, and so there's true suction feeders, and then there's raptorial suction feeders that grab the prey and then use suction, as opposed to just suction the entire way. So there's a bit more that could be done with this, but this was kind of the first look at, it was a phylogenetic context to look at suction feeding. These characters really aren't so important, but just to show you that that suction feeding goes way back. And actually, that's not so surprising, because when you think about it, any animal, any vertebrate that's in the water is going to have to generate suction to be able to feed. So not a surprising result, but definitely a confirmation. And then there was what she was also looking at is, there was a previous paper where did not use a phylogenetic approach, and then said, well, since we've got suction feeding and all these different groups of tooth odontocetes, gee whiz, suction feeding must have evolved independently in all these groups. And because she was able to look at a broader context, she was able to say, well, that's not really true. Suction feeding really evolved probably in a general strategy. It evolved once, kind of at the base of the tree, and maybe even involved certainly some of the stem whales, the archaeocetes here as well, and then filter feeded being unique to mystocetes. So again, using a phylogenetic framework, we can hypothesize and confirm that suction feeding as a general strategy evolved once. This is some more work by Tom Demeray and a colleague of his, and this is on Herpetocetus, which is an extinct taxon, and they did some muscle reconstructions. And this is, their idea is that based on these muscle reconstructions, and they took the fossil mandible and the skull, and they pulled the jaws apart, and they saw what would be the limits of gape, right? And so they were able to show that, well, this Herpetocetus, this extinct whale, wasn't really able to gape all that widely. And so it also shows a number of anatomical characters that support the fact that it was a lateral suction feeder, similar to gray whale. And of course, there's a lot more that needs to be done with all of this in terms of, you can imagine the biomechanics that could be done with this. This is an artist's reconstruction of, well, maybe this Herpetocetus, this type of cetothere, really had lateral suction feeding. So it's more specialized than the generalized strategy that we think is ancestral for the whole group. Okay, this is a little bit busy, but the point of it all is just to sort of show you through these different transformations. We think some of the tooth mystocetes had teeth and baleen, and then the ones that are baleen-bearing lost functional teeth. Within this extinct group, the cetotheres, we're just starting to look at some of their feeding characters, but they start to show, bowing of the jaws are coming out. Within the crown group, we see a whole slurry of other characters. These ventral throat grooves, again, allowing the mouth to expand to great proportions, take in large amounts of water and zooplankton. The evolutionary scenario would say, well, suction feeding, that's the most generalized strategy, probably single prey feeding, and then perhaps we had bulk filter feeders. So within these, what we can say is lateral suction feeding probably, at least if we map it out on the tree, probably evolved at least twice, right? It's seen in the cetotheres, the extinct group, some of the extinct guys, as well as the gray whales. And then in case anybody's interested in whales, there's really a whole lot you might think these are large animals, we know a lot about them. We do know a lot about them, but we know less about the anatomy, and as I've told you, we know even less about the systematics. But this is one area that has really not been studied at all, is how right whales feed. Right whales and pygmy right whale. So a couple of some future work that we're doing. One, we're continuing to phylogenetic work. But one thing, another area that my lab is not going to do, but John Gatesy and one of our colleagues, Hans Tewison, is looking at the genetics of baleen. And it's, like I said, difficult to get the fresh tissue data that you need in order to be able to do this. Look for the candidate genes. And it's been quite difficult, just because you really have to go when they're, you have to participate in a bowhead hunt, I think, to get the baleen, because it's got to be fresh. So, but I think finding the candidate genes has been more difficult, but that's could be a very lucrative area of research, I think. Also, there's more to be done on the development of baleen itself. We've looked at gray whale in some, a little bit, but there's a lot, lots of other species to look at. We obviously need to look at the palate vasculature in other species, do more vessel injection. And then one area that I've recently gotten involved with is because we got a laser surface scanner in the lab. So we've started, kind of thinking, well, we should be able to do more with the morphometrics of the skull. And so these are large animals. The problem is the laser scanner takes a long time to scan a skull, but I have a grad student now that's looking at really getting some data on growth of the skull, and also looking at certain feeding anatomy characters, so that maybe we can start to approach feeding mechanics. So almost at the end here. Here I am with, yes, I dye my hair white now, so that's somebody actually asked me that. And I was like, are you kidding, really? And so we've had very, very fortunate to have NSF funding. I had a Fulbright, lots of individuals to thank for collections, access, and all of these master's students and undergrads have been involved in the lab over the years. And then, of course, I have to put my promo in here. It is shameless self-promotion. This is coming out in another year, and this is a book that I wrote for non-majors that kind of gets at lots of aspects of whale biology and kind of uses them to explain principles of evolution. So with that, I will take questions. That's another slide. So thank you. Oh, I ended on time. Yes?
So Annalisa, really not much is known about what genes are involved in the production of baleen?
No.
Really? It's complete mystery?
Well, I'm going to a meeting next week that Hans is going to be at, and so he's going to talk a little bit about that. He's working on bowhead whale. But yes, it's kind of an open question.
Whales are still hunted, right?
Yes. Well, that's where he works, is doing the bowhead. Those are Inuit hunts that are up in the Arctic. But yes, it's true. You could probably, the Japanese still hunt minkies. Right. So I've been to Japan, and the problem is that they they're kind of low key on that stuff because they still do scientific whaling. So yeah, that's the problem. Is if you could. They don't have they don't have collections of the osteology, but it's worth asking them about. But it's always dicey because then if they want a collaborator from the U.S., then you can't really go to NSF and say, "Will you be willing to support this kind of research that's scientific?" Yeah, exactly. It doesn't work. So, but there certainly are. I've seen a lot of fetal material there, so you could do some other stuff with it. Maybe not the gene stuff, but you could do some development. But if you want to do gene expression, I mean, yeah, you'd have.
It seems like you'd have to get developing here in whale.
Right. That's my understanding.
But there's some genomes out there, right?
There are some.
Unusual about the whale.
I don't know any. I know more about the toothed whale gene that's been done, the Terciops. I mean, I think there are different people working on parts of it, but I don't know that they have a complete genome for, or do they, for humpback? Maybe? There's a minke, it's been done. Yeah, well, they've looked at certain, I don't know, certain other genes and positive selection and olfaction, for example. I know they've looked at that, some of the other genes for some of the other special senses, but I don't know that there's anything that's come out of that pertains to the baleen itself that I'm aware of. So there you go. See? Something to do.
Yeah, Brad?
Yeah, that's great. So I've always assumed, I guess, that with baleen whales eating, it was sort of more about gram and less about suction, if you make that distinction.
Uh-huh.
Make that distinction. And yet I gather from this, your take is that it's all about suction and not about.
Well, I wouldn't want to I certainly think it's a combination of things. And most of our work, though, really is not at the biomechanics of it, I'd have to say. I mean, we're looking at some of these gross anatomical structures, but it's not within some of the work that, like, a colleague of mine, I guess. You know Jeremy Goldbogen, maybe. Now at Stanford, anyway, he's done, done a lot of the digital tag work, and now he's looking at, the biomechanics. So I think it's going to, it's going to have to be different kinds of data that are kind of, coming together to tell the story. But I think it's both. I wouldn't want to say it's more suction than it is, ram feeding.
Because do they feed when they're not actively moving forward? I mean, do they, do the sort of standard whole feeding baleen whales? Do they ever just sit there and feed, or is it always forward?
For the balanopterids, they're rapidly moving through the water. But as I say, they've got some really cool, they've got these tags, and they've got the tag data, also the tags and acoustic data, so now they can tell if they're making, sounding. These guys don't echolocate, but in some cases they still, well, the question really is, how do they find the prey? And this is some other work that Hans Tewesen is doing, again with the bowhead. He's done some work on bowhead anatomy, olfactory anatomy, and shown that the olfactory system in odontocetes, in the toothed whales, is non-existent pretty much. It's very, very reduced, mostly absent. I guess it's a little bit of it's developed as in the fetal stages, but then it disappears. And in mysticetes, they have it's a reduced olfactory system. So the idea is that, and there's some suggestion that they use smell in some way, perhaps to locate plankton blooms, and they can sense some of the chemicals that are in the water. So that's been a new idea. It hasn't really been followed up, but there's some other interesting line of research. Yes.
I have two questions.
Uh-huh.
One of which will be completely off the wall. I'll save that for the second one.
Thank you. I guess.
The first one is when thinking about transitions, evolutionary transitions like
Uh-huh.
this one, it always occurs to me that at every stage there has to be some selective advantage to continuing
Right.
in terms of the development of things like baleen.
Uh-huh.
Okay. What do you see these intermediate forms as being and having an advantage as compared with other things that also were feeding on, say,
Yeah.
small fish or medium-sized mollusks or whatever?
Well, again, I think in that Alfaro paper, he's not here, is he? Okay. Well, anyway, they did. Graham Slater talked about partitioning out the niches. So some of nobody really looked at the baleen whales. I mean, mostly he looked at toothed whales, a little bit with baleen, but I think he just grouped them as all kind of zooplankton feeders.
That's all circumstantial evidence.
Yeah, I don't know. Nobody's really.
Anybody expand their niche? I mean, just because
Well, that's
we
what
had- Yeah.
you're waving your hands, though. It's an evolutionary just so story.
Well, there's a lot of food out there. I mean, they're partitioning it up in some way, but how do nobody's.
I don't see what the driver is, though, frankly, in terms of.
Well, high productivity.
That's
That's kind of what. There it is. I got to use it. This is a slide just to show the increase in. This is sort of an evolutionary driver.
I didn't see that slide.
Okay, well, there you go. So, but the question for me is, this is sort of looking at diatoms as productivity measures, right? Which is, we know that's the case. But I mean, nobody if we really want to get at baleen whales, I would think we'd want to look at krill, but I don't know how you could look at the fossil record of krill. I don't know what the fossil record is. I don't know. I mean, that would be a way to get a little bit closer to it, I think, than this, but I'm not aware of any fossil records, so I don't know. So that's,
So let me
But
ask
you could see this huge diversification.
Right.
Competition could be a driver.
That's a possibility.
yeah. Well, there's peaks of when diversification, major times of diversification.
Usually evolutionary pulls and pushes and so
Yes.
of those kinds.
Well, there's some really interesting Alfaro kind of clay diversification questions, but some of it you can't do, or difficult to do without having a better phylogeny. And until the phylogeny is worked out, some of that just, you can only do generalizations. But I certainly think you could test some of this by looking at evolution within a clade, within different clades.
So let me ask you the
Okay, here comes the off the wall. All right.
And that is that this open ocean niche that
Yeah.
filter feeders are occupying obviously has been around
Yes.
since the beginning of the oceans. So it's about
Yeah.
2.5 billion years. And there have been a series of faunas
Uh-huh.
over time that have occupied that.
Mm-hmm.
Now, the one that's relatively most recent, prior to the evolution of the whales, was back in the Mesozoic.
Mhm.
All of the large marine reptiles, the Mosasaurs and the Ichthyosaurs and Plesiosaurs. Is there any sign of anything like this?
Well, I showed you the.
Mesozoic reptiles.
Oh, no.
Christine actually wrote a paper about this.
Oh, she did.
Yeah, Christine Janice.
Okay, well, I'm.
Why no suction feeders in the Mesozoic? And I think, it's been a while, but.
Huh, I haven't seen that.
Paper. I think it might have to do with the evolution of the secondary palate. We should
Oh.
close off the back of the mouth in order to create suction.
Uh-huh, yeah.
Or to really create big suction, I guess, for these guys. I don't know, so I'll find it for you.
Yeah, find it for me too.
I'll find it
I didn't
for you.
I didn't know about that.
Remember it was published, but yeah.
I don't know about that.
You always, yeah, it was an interesting paper.
Yeah. Yeah. Interesting.
Well, we forget that right in the Mesozoic that there's all those big fish around.
Yeah, exactly. Well, yeah, there's the gill rakers and the cartilaginous
They've
sharks.
always been there.
Yeah.
Back since the
No, no.
Anyway.
Well,
Yeah. Yeah.
Mesozoic.
Right. Certainly.
Yeah.
All right. So thank you.
One other thing with the artiac, when you got me thinking about the.
Oh, okay, the palate. Yeah, so what do you think?
Do they have teeth that are resorbed as juveniles? Do you know? No, they
I don't think so.
I don't think so. We would
I don't
know
think
that.
so. I think we would know that. Yeah.
There'd probably be an occasional mutant.
Yeah, but they have their whole, I don't know about the epidermal, yeah, exactly. I don't know. But I mean, I think that's obviously going to be closer than looking at platypuses. Although the platypus thing, there was something kind of, it was an analogy. What can I say? All right. Thanks.
Thank you.